Alright, so this is week 2, and we are doing the tissue level of organization. We started at the cellular level. Frankly, we skipped chemical, and we're going straight up to tissues. So a tissue is a collection of cells that work together towards a common function. And there are 4 main types of tissue. Everything in your body can be categorized into one of these 4 tissue types: epithelial, connective, muscle, or nervous tissue. Those are the only types of tissue that you are made of. 4 types of Legos, so to speak. Since they have these only 4 categories, there are different numbers of types of tissues that go into these 4 categories. Epithelial is going to be our most dense category. We have the most options for things that line and cover things. We're going to notice that, we're going to feel like we take forever on epithelial, and you'll be like, wow, there's 3 more types to go. This is going to take forever. That's not accurate. It's going to get shorter and shorter. And by the time we get to nervous tissue, there's only nervous tissue. There's just one thing. There's 2 types of cells in one tissue, and we're done. So it'll speed up as we move along. Brief notes on tissue changing and age. Okay, we did that already. Tissues vary in structure, function, and the content of the extracellular matrix. Again, with their structure-function relationships, part of that pattern recognition that gets people good at histology is saying there is a connection between the way the cells are arranged and the job they're built to do. So we're going to do as much of tying in those ideas of structure-function relationships today as we can. Epithelial tissues, in general, they're going to be lining something. When you look at a slide that has epithelial tissue, you're going to see these white spaces. If there's a white space, that means it's either facing the environment or it's facing a space within the body, such as the inside of the stomach is a space, right? The inside of blood vessels are spaces. Every space is going to be lined by something, and that lining is the epithelium. That's the definition of an epithelium. They all share some very important characteristics. Characteristics like being, let's see, there we go. Cellularity of, it's always cells. There's nothing outside of the cells. This is going to contrast it to something like connective tissue, where you have cells and a whole bunch of stuff in between the spaces between the cells. So we're going to call that space between the cells and the stuff in it the extracellular matrix. Epithelial tissues will not have extracellular matrix. It's all going to be cells for the entirety of that epithelial tissue. Because there's going to be a side that's connected to the rest of you and a side that is free to a space, we're going to call those cells polar. There's going to have polarity. They're going to have a top and a bottom. We're going to call that top apical. We're going to call that bottom basal. Those are just vocab words you're going to see eventually. Later on, we're going to use those terms occasionally to describe how things flow through epithelia if we have to have, say, gas exchange through a surface or something secreted from that surface, other things that epithelia can do. Then we'll say, okay, that's going to go from the apical surface to the basal surface, and it's going to diffuse in this direction. So that's long-term vocab for you. All epithelia are going to have a basement membrane, which means that there's a place where they attach to the rest of the tissue that separates them from the rest of the tissue. And beyond that basement membrane, they are avascular. What does the prefix a mean? Without and vascular? There's no blood vessels, so there will be no blood vessels in any epithelial tissue. Now, what are blood vessels there to do? Why do we have blood vessels at all? So you need blood flow because it's bringing something to your tissue. What's it bringing? Life, sure. Oxygen, nutrients is what it's bringing. So that means that the further away you get from that basement membrane, the less nutrition and oxygen you have for those cells. So if you have a relatively thin epithelium, that's not an issue. But if you get a thicker epithelium, that means the cells far away from the basement membrane, the cells that are way over here, those are going to be dying. Those are starved. They have no oxygen. So the number of layers that an epithelium has is going to be very, very important to us. In fact, it's going to be part of how we name our epithelia. Epithelia generally are innervated. They will have nerves that can go past that basement membrane, so we can detect those paper cuts. Aren't we so grateful for that? And high regeneration capacity. It is part of the job of lining in times is going to be protective. This means it's going to be a vulnerable surface to everything from, swallowing a Dorito or, anything that can travel through any of those spaces that these things are lining. It's very plausible they'll get cut. Or if it's, for example, in the stomach lining, that acid that's in the stomach is going to eat away at it. So these cells are very capable of mitosis and of healing and of regenerating. So high regeneration capacity. So here's the general structure. We're going to have general tissue down here. We're going to have a basement membrane separating the epithelium from the general tissue. This will probably be connective tissue down here, statistically. And then we're going to have our basal and apical surfaces and lateral surfaces connecting adjacent cells. So again, form and function, physical protection. The best way to get physical protection from an epithelium is going to have a lot of layers, or to have some kind of cell-to-cell connection that's going to be very strong or protective. Selective permeability. Let's say you're in the digestive system and you've eaten a hamburger and you need to get those proteins, fats, carbohydrates from the lumen, from the inside of your gut tube, from your small intestine, into your bloodstream. That's going to allow for permeability of those things. Proteins, fats, and carbs are going to be permeable through that digestive epithelium. But it's not going to let anything in and out, right? We don't just let everything into the environment. So for your skin, for example, it's a selective barrier. It's not going to just let bacteria through. You're going to have bacteria on the surface of your skin, but it's going to have a lot of layers, so they're not going to get through those layers into your body itself, which is good. We don't like that. Secretion, all of our glands are derived from epithelium, every single gland. Especially the sweat glands is what we're going to see a lot of today. Sweat glands, for example, and that'll help us secrete and get rid of things or just create nutrients or things that our skin needs, for example. And then sensation, because it is innervated, all of our special senses are epithelia derived as well. So you'll have, for example, an olfactory epithelium in your nasal cavity that allows you to smell. So again, that basement membrane separates other tissues from epithelia, and we will be avascular beyond that. It's generally connective tissue below. Again, that's a different category for tissues. Different epithelia will have different intercellular junctions. And I keep meaning to actually get out my sewing machine and sew a couple of pieces of fabric together to demonstrate these. This is going to be on your worksheet today. This is where you're going to say 4 major types of cell-to-cell junctions, and each of them are going to be located in different epithelia. They're going to have different jobs to do. They're going to have different roles. So our first one is tight junctions. And luckily the name tells you a lot about it. The idea with a tight junction is if you were to get your sewing machine out, it would be a very tight stitch that's repeating and overlapping with other stitches. And the idea with a tight junction is you're not going to be able to stick your hand between these two sheets that you've sewn together here. So with a tight junction, if I'm a bacterium or a larger molecule that's hanging out right here, and I want to go from here to here, I can't. I'm going to be stuck here. I'm going to go brink, 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 and I'm going to hit that tight junction, and I'm not going to be able to get through it because it's tight. It's tightly woven, it's tightly knit, it's tightly connected. So I think I've got, yeah, tight junctions. They're going to prevent molecules from traveling between epithelial cells. They're going to be gatekeepers between the external and internal environments. So if I'm a molecule trying to get from here to here, if there isn't some cellular mechanism like endocytosis, exocytosis, some kind of protein channel here that is for me, I'm going to just be out here. And that's probably good, right? If we are not selecting for it, we want to be protected from it. Our next junction is going to be adhering junctions. Now adhering junctions, let's see, we call these purse strings. If you can remember nothing else, remember purse strings. They're going to stabilize the apical surface. So there is generally specific locations for these, either apical or basal. And those tight junctions tend to be more apical. The adhering junctions, sometimes you'll see the word adherens junctions if you look in literature, same thing. They tend to go circumferentially around in 3 dimensions the entire cell and adhere it to every other cell that is on a lateral surface, and it's just going to stabilize it. So it's not going to be a selective barrier like tight junctions, and it's not going to be as quite of a protection from shearing as desmosomes that we'll get to in a minute. It's just going to be stabilizing it. It's going to be encompassing the whole thing, like if it was in the middle of a group hug kind of thing. Like it's harder to jostle us if we're all in a group together, right? So third is a desmosome, and I compare this to a rivet. So they're going to be spot connections. This was a stabilized purse string all the way around that apical surface. A desmosome is in one spot, connecting the other cell just at one spot. So again, if you're a molecule trying to get around this one spot that's connected, that's easy enough. And it's not so much a group hug as like we're doing what's that game where you're all chained up and people try to break the chain? Red Rover. It's more like Red Rover, right? The idea is we've got to hold on to each other as strong as possible, and if people try to tear us away from each other, it's going to be very difficult. So a desmosome is a rivet that is going to make it a very strong connection between cells. It would be hard for them to be torn away from each other. But it's not going to prevent any of the other things that we've talked about. It's not going to prevent molecules from getting in or out, or bacteria, or anything like that. So we're going to see desmosomes in cells that need to be very strong with each other. Like the surface of your skin has a specialized sort of cell-to-cell connection, much like a desmosome. That means that if your skin's coming off, it's flaking off altogether, right? We've all been flaky at some point. Those are large sheets of cells coming off that have died because they're far away from the vascular supply, and they're all connected with desmosomes. Finally, gap junctions. And again, the name tells you a lot about what's going on. A gap junction is a tunnel from one cell to the next. This is cell 1, over here is cell 2. There's the cytosol of cell 2 over here, cytosol of cell 1 over here, and it's just a tube, and it's going to allow for the passage of ions especially. They're going to share that fluid content of their cells. It is continuous when there are gap junctions, which means if this cell suddenly gets a bunch of calcium, then this cell also suddenly gets a bunch of calcium at the exact same time. It can just go straight through this channel. So gap junctions are something you're going to see in cells that need to communicate very quickly. Okay, any questions about those cell-to-cell proteins? Okay. What do adherence junctions do? Yes, purse strings that stabilize apical structure. Perfect. What do tight junctions do? What's that? Gatekeepers prevent molecules from getting through the epithelium. Excellent. What's a desmosome going to do for you? Rivets, making a strong connection. Absolutely. And what's a gap junction going to do for you? Absolutely, yes. It's going to be like a tunnel, just as Jenny said. It's going to be like a tunnel that allows for quick communication and sharing of ions across cytosol. Excellent. Okay, our naming conventions for epithelia are going to be really cool. We're going to start by the number of layers of cells and the shapes of cells. In fact, when you are trying to figure out what type of epithelium you're looking at, what you're going to do is you're going to first look at the general structure of it. You're going to ask how many layers are there, and you're going to ask yourself what shape those cells are in, and that's going to be pretty much half of the name of your epithelium right there. So if it's only one layer, we're going to call it a simple epithelium. One layer, simple. If there's anything other than one layer above that basement membrane, we're going to call it stratified. So even starting from 2 layers, that's a stratified epithelium already. Generally, when you see stratified, there's only a couple of epithelia that only have 2 layers. Most of them are going to have many, many layers like this. So there's a simple epithelium, there's a stratified epithelium. 2 or more layers, absolutely. The second naming convention, which is also our second classification strategy, is shape. If it is flattened, we call it squamous. So if you only had 1 layer of squamous epithelium, you would call that a simple squamous epithelium. And you're done. That was that first epithelium we're going to see. If it's only 1 layer of a cuboidal cell, and a cuboidal cell, it's not always exactly going to be cube-shaped. It's going to be as tall as it is wide. So circular is also an option there. Cuboidal cells, if you only have 1 layer of cuboidal cells, what would you call that? Simple cuboidal, perfect. And then if your cells are taller than they are wide, from the basal to apical surface, they are taller than they are wide to the lateral surfaces, then we'll call that columnar. So if it's only 1 layer of columnar cells, what do we call that? Simple columnar. Yep, so you just learned your first 3 epithelia. Those are all types of epithelia. What if you had more than 1 layer of cuboidal cells? Stratified cuboidal. 1 more or 2 or more layers of columnar cells. Stratified columnar. There's only 1 place that has stratified columnar. What's that? I want to say malurethra, but I'll have to check that. So, there's 1 more curveball here, and that there is such a thing as a pseudostratified epithelium. I'm going to write this up on the board. Sorry to... Actually, I might pause this while I write this up on the board for our listeners. Okay. So here are all of your options for types of epithelium. Simple squamous epithelium, just 1 layer of very flat cells. Simple cuboidal, 1 layer of cells that are as tall as they are wide. For simple columnar, you also have a specialized option. It can be ciliated or non-ciliated. What are cilia? Yeah, movement. Yes, movement. So microtubules, part of the cytoskeleton, helps with movement, make up your cilia. So cilia will generally only be found on columnar epithelia, sometimes pseudostratified, but we're going to focus on columnar. Columnar. So cilia will be found on columnar epithelia. Yes? Yes. So ciliated and non-ciliated, you know, I would be getting ahead of myself if I said the next point. I'm going to stop. Oh yeah, sure. Do you want to pass them out? Thank you. Okay. Now, for stratified squamous, you also have one more option. Keratinized or non-keratinized. What keratinized means is, and again, this is stratified squamous. What did we say about getting farther and farther away from that basement membrane? Dead cells. So keratinized epithelia, keratinized cells mean they're far enough away from the basement membrane that they've died. That is what keratinized means. So in a keratinized stratified squamous epithelium, we have many, many layers. We have dead layers on the top. Generally, they're going to serve some kind of purpose. So your skin is a keratinized stratified squamous epithelium. That keratinized layer, that's what sloughs off when you exfoliate. That's what flakes off when you're dry.
So would that be still considered high regeneration?
It's still high regeneration capacity because you still have many layers of that epithelium, and that part that's close to that basement membrane has very healthy cells that continue to regenerate. Yes, very much so. Stratified squamous non-keratinized epithelium is not going to slough off. So we're still going to have it in areas that need a lot of layers to be physically protective, but we're not going to have that flaking. And generally when we see non-keratinized stratified squamous epithelium, we're going to be really glad that it's non-keratinized. You'll see what I mean later. And then we're back to our simple stuff. Stratified cuboidal, stratified columnar, pseudostratified columnar, and finally, another weirdo over here is transitional. Only in one place, urinary bladder. If you're looking at transitional epithelium, you're going to be in the urinary bladder. Possibly in the ureters, I think. So the thing about transitional epithelium, what makes it transitional is your bladder has more than one state it's in. It can be empty or it can be full. And when it's empty, it's going to be kind of loose, and those cells that are on top are going to be kind of rounded. It is a stratified epithelium. There's many layers because it's going to be stretching out. It's got to be protected for that. When it's relaxed, the cells are going to be rounded. When it's distended and full of urine, they're going to be flattened out. So it transforms from a sort of cuboidal shape to a squamous shape. That makes it transitional. That's the transition. So we're only talking about vocab right now. We've only used words. Let's go ahead and attach some pictures to these ideas now. Any questions before I move forward? Okay. So one of the downsides of using all textbook images for these is these are probably not the images that I would have used in some cases. This is definitely not the image I would have chosen for a simple squamous epithelium, especially starting out. The key to understanding that this is a simple squamous epithelium is that we're only talking about this one layer right here. We're not talking about any of this stuff, and we're not talking about this. So if it were up to me, I would have had more white space up here. I do like having this cartoon version down here because it is highlighting this one layer of cells that we're actually talking about. This is why I keep the cartoon version. So as you're studying that, make sure that you ignore all of this stuff. There are places in your body where you can go that just have that simple squamous epithelium without a bunch of connective tissue right here, like in your lungs, for example. That's probably what I would have used, but this is your image for simple squamous. Hello. Any questions about that? Yes?
Practical, what would
So for the most part, oh, that's actually a great question. For lab practical purposes, there are a couple of ways this could happen. For the most part, it's going to be ID this tissue. ID this tissue, ID this tissue, ID this tissue. If we get into tissues with really strong function structure relationships, I might ask you like a one-word answer. What is the purpose of this tissue? Is it protective? Is it going to allow for gas exchange? Something like that. Really brief, something that's going to fit on a line that big. And only if it's very clear, only if it's something that I've talked about a lot in this lecture. That's what I would put on there. As for identifying cells, there are probably only a couple of tissues where I would say, I want you to be able to identify that this is this cell or this is that. So for example, neural tissue, there's only the one type of tissue, and there's only two types of cells. So probably learning what those two cells are called would be a good idea for that. But I'll try to point that out as much as possible, and please do keep asking me those questions. All right. Yeah, lab practical happens first, and then you get about 10 minutes of like walking around time, and then we do the lecture exam next week. Okay. Simple cuboidal epithelium. This is pretty much a great picture to use for this. It's an unusual stain, but you can clearly see the basement membrane here, thanks to this great stain. Here it is up here, basement membrane. And you can clearly see, and these are rather cube-shaped too. So these are kidney tubules. You won't see these again until we get to the end of AMP2. But there's your simple cuboidal epithelium. So again, notice that the plasma membranes aren't as easy to see as the nuclei. Usually those nuclei are going to be these dark purple things. In squamous, they'll be kind of flattened as well. The nuclei are always going to be just nice circles for the most part, are going to be nice circles that are going to be a little bit darker than the rest of the cell.
Can I ask real quick on the page right before on the single Squamous epithelium?
Yep.
So the single layer is on the top that we're...
Yes, the single layer on the top.
What is the bottom?
That looks like a simple cuboidal.
Okay.
Yep. This is the amnion. This is a stage of embryological development. So actual developing human cells are, I believe, down here. And this is going to be part of the yolk sac is what I believe we're going to call that. I could be getting that wrong, though. Sorry if there's any embryology specialists that find this on YouTube and are shaking their heads right now. Okay. Which one? Okay. Yes, absolutely. So I'll point it out physically on here, then I'll walk over there and it'll go quiet on YouTube for a second. So this is tissue that is separate from the epithelium, and that's the nice thing about having the medical illustration is that they've highlighted the epithelium. This is the lumen space, so you've actually got two layers of epithelium here. You've got a layer of simple cuboidal epithelium here, and you've got a layer of simple cuboidal epithelium here. So I'll go up here and do this. So what you're actually in is a kidney tubule. There's fluid in this space right here. It's urine, this space right here. This is one cell, this is one cell, this is one cell. So they're all adjacent to each other, making this lining. And this blue stained area, that's your basement membrane. So this is a simple cuboidal epithelium. This is also another basement membrane. That's a cell, that's a cell, that's a cell, that's a cell, that's another simple cuboidal epithelium. Okay. Does that work a little bit better? Yes. And this structurally, this image is similar in that we've got one lumen space and we've got two epithelia, one on that orientation, one in that orientation. So once again, we've got a basement membrane here. This has a goblet cell. So a goblet cell is going to be a cell that secretes mucus. We're going to see this in a few structures. Lots of mucus in your digestive mucosa, so that's not that surprising. So note that it's hard to tell in this actual histology image where one cell starts and the next begins, right? So you're kind of looking at the blue or at the dark purple dots, the dark purple circles, and saying, okay, those are my nuclei right there, and they're more or less aligned. Goblet cells create mucus, yes. So see how those nuclei are more or less aligned. Now here's a little bit of something that's a challenge. Remember that microvilli and cilia can kind of look a little similar? So this is non-ciliated. We're about to look at a ciliated simple columnar epithelium. For a novice histologist, and even for an experienced histologist, I'm not going to lie, it can be hard to differentiate a brush border made of microvilli like you see here. See how it's just kind of a little hazy right there? Versus ciliated, which is going to be a little bit longer, a little bit brushier. That's a bad word to use because we just use brush border for microvilli. A little bit more spiky right there, right? Again, that's why I'm only using these images for your histology, for your lab practical. I'm not going to change it up on you to a microvilli brush border that looks a little bit spikier than that. It's just going to be this image. That's why I do that. So if there are microvilli, it is not considered ciliated, even though it may have a brush border. Generally, this is a smoother structure, so these are good slides to demonstrate the difference between non-ciliated and ciliated. Yes? Yes, I will show you this entire image. This may be cropped or blocked out if it makes it too obvious what it is. I have the ability to cover that. But so generally you will assume that it'll be cropped to a rectangle, and you may not be able to see what organ it's coming from. But you will have the actual histology image, and you will have the medical illustration image. I never block out the medical illustration. These words won't be here, though. Don't expect the words to be there, just like it's in your blank images that you have. Okay, questions? Yes?
So we're only labeling what the medical scientists will not label.
No, I'm not going to ask you individual words from here. I would say ID tissue. In very, very rare instances, if it's something where I've said that there's a really, really strong structure-function or organ relationship, I could maybe say one place that would have this. I probably wouldn't for kidney tubules. You're not going to understand kidney tubules for a really long time. Yeah. Okay. Ciliated simple columnar. This image kind of flies in the face of one thing I just said, and that is in the actual histology image, you can look for those nuclei to be more or less lined up. Over here, those nuclei are more or less lined up. Over here, it gets a little bit messy. It's still simple columnar, especially in the cartoon version and the illustration version. You can really tell that those cells are all adhered to the basement membrane. They are all longer than they are wide, and they are relatively orderly. So in histology terms, this is an orderly appearance. In, normal person terms, maybe it's not so well aligned. I would go by this over here. If you're trying to get that, it's not pseudostratified, it's simple columnar. Therefore, the nuclei are aligned with each other. I would just look over here and sort of circle that mentally or physically on your flashcards, whatever tool you're making to study these. So you do have to write this whole thing on your lab practical. You have to say it's a ciliated, simple, columnar epithelium. So the location for this is the uterine tubes is a really good place for this. Those cilia are going to move that egg, that oocyte, along through the fallopian tube, the uterine tube. Questions? Moving out of simple, we're going to start going to stratified. First, we're going to see non-keratinized stratified squamous epithelium. So with stratified epithelium, The hardest part for this as a newbie is probably going to be identifying where the basement membrane is without a line showing you where the basement membrane is. The basement membrane is down here. This is the division between connective tissue down here, and all of this is epithelium from here to here. So you can see very, very many purple dots indicating very, very many nuclei of very, very many cells. And we go by the shape of the top cells. So sometimes if you look at this bottom row of cells, it doesn't look squamous, it looks a little bit cuboidal, but they flatten as they go towards this apical surface. You guys see that? The key is that they're flat up here, so that is still squamous. So again, pattern recognition, this is the basement membrane. We are talking about very, very many layers of flat cells. We are going to have to go back and forth. Keratinized versus non-keratinized. On this picture, the basement membrane is down here. It is a different stain. That's why it is red and blue. This would not be the picture I would choose for this. Basement membrane's here. You've got a bunch of living cells, many, many flattened layers of living cells, and then you've got many, many layers of dead cells up here. Notice there are no dark purple dots up here. That's all dead. If the nucleus is gone, it's gone in the skin. So non-keratinized would be found in a place such as the vagina. It's also found in the esophagus, places that can be exposed to quite a bit of friction and need a lot of protection. And thank goodness this is non-keratinized, otherwise we would be sloughing skin from our vaginas, and that would be very unpleasant. So, yes, I know, it's gross. It's gross to think about. Okay. So again, this is keratinized, stratified squamous epithelium. Again, get used to writing the whole thing. I would recommend actually physically writing this out while you test your cells. Make sure you write the whole thing. Keratinized, stratified, squamous epithelium. That's, if you have half of this information, if you tell me it's a squamous epithelium, that's approximately half a point. If you get it wrong, then it's wrong. If you give me stratified squamous epithelium, I still only give you half a point because keratinized is part of it. Questions about those? So the basement membrane's down here, or down here, and then you look for the dark purple dots, which again, this is a different stain, so it looks really different. There's many, 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 many layers. So all of these, this is a layer of cells, 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 this is a layer, that's there, 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 there are this many layers of dead cells in here. This is a lot. This is what we would call thick skin that we learned about in the integumentary system. This would be like the soles of your feet, palms of your hands that have many, many layers. Like, have you ever stuck a needle in the skin of your hand and it's fine? That's the needles in here. No, the lab practical will be projected up here. It will be in color. But the lab practical images that you print will be in black and white. So unless you've got access to a color printer, then there is that extra step. Color printers are good. Color printing is a good idea for this. I don't want to add cost on to you guys, but maybe. Yeah. Yeah. Maybe like collectively go to Kinko's and get it done. Okay. Stratified cuboidal. Again, thank goodness the medical illustration is there. So the basement membrane is here. And you see those purple dots? You've got 1, 2, 1, 2, 1, 2, 1, 2. By the time you've got more than one layer, we're already calling it stratified. Does that visually make sense? Okay. Everyone in the back? Give me a thumbs up or a thumbs down. Okay. Thank you. Stratified columnar. Here's the basement membrane. Here's the basement membrane. So this one, it can be very, very hard to differentiate from pseudostratified. That's our next slide. So again, we are looking for those darker purple dots. This is that red and blue stain that I'm not a huge fan of for you guys. So there's the male urethra. I was right about that. Stratified columnar is exclusive to the male urethra. That's the only tissue that has it, as far as I know. There, I think, some glands that do. I always say it's only in this place, and there's always a gland that has it too. But for your purposes, male urethra. Stratified columnar. So they are taller than they are wide, especially on that top layer. Remember, we mostly go by the top layer, the apical layer. That basal layer kind of gets a little squished. But they are taller than they are wide on the apical layer, and they do have a relatively orderly appearance of their nuclei. I say relatively. Now we're going to contrast that with pseudostratified columnar. So again, this is why I have the medical illustration version. You can see how if you were just looking at these, you would be looking for the dark purple dots, and you would be seeing them in many locations, some of them higher up, some of them were apical, some of them were basal, and you might think to yourself, okay, that looks stratified in some way. But with the medical illustration, we could see the plasma membranes are, again, this is a three-dimensional thing, so we're not seeing every piece of every cell. Some of these are cut at angles. They are all adhering to that basement membrane. And this one happens to be ciliated. So you're never going to see cilia on a stratified epithelium. It's only in simple columnar epithelium. So that cilia is your second evidence that you're not dealing with a stratified epithelium. Okay, we'll do transitional epithelium, then we will stop and go on our first break. Questions about pseudostratified versus stratified columnar versus probably simple columnar is what you would need to compare that to most. So take a look at your image for simple columnar. Remember, this is orderly, at least over here. That's going to be your reference point. Versus this here, where the nuclei are at different levels, but these are all cells that are as wide as they are, narrower than they are tall. And again, you've got goblet cells here. That's what these white spaces are. Is more goblet cells secreting more mucus. Surprise, you have mucus in your nasal cavity. Yes, Tamara.
You may have already said this. So on the lab practical, when we give us these images, do we have to, like the top or the nasal cavity lining, do we have to label that too?
No.
Okay.
So it's going to be very specific questions. Most of them are just going to be ID this tissue. Rarely it will be ID a cell, only if I've really, really, really emphasized it and danced around and gone like this a whole bunch of times. Whee! Occasionally where it would be found, also if I've been like, wow, isn't that so important that we have that there?
Okay.
Yeah. Again, I dance around a lot if it's really important. All right, finally, transitional epithelium. The apical cell shape changes depending on whether the tissue is stretched or relaxed. So this is the apical surface, and here it's rounded, which makes sense from a histology perspective because it's going to be hard to fix and prepare a urinary bladder that is still full of urine. Part of the fixing and preparing of slides involves, that tissue is dead, so it's not going to be full of urine at that point. It's going to be difficult they would have to find a way to microscopically stretch it to show you what it's going to look like squamous. So the basement membrane is down here. We do have multiple layers of cells. I think the best way to say that this is transitional and not stratified squamous, since that is the biggest stratified tissue that you find in the most places, is the rounded surface on the top. You'll definitely not see that rounded surface on the top for something like non-keratinized stratified squamous epithelium or keratinized stratified squamous epithelium. Those are both very flat, whereas this is rounded. So you start to see the pattern recognition that goes into this. Even just looking at one slide example for each of these types of tissue, imagine somebody just throws a random slide at you, you don't know what organ you're in, you don't know what part of the body you're in, all you see is that there's a white space and you kind of have to work your way backwards from there. You have to try to find that basement membrane. In this picture, it is not a stained basement membrane. It is hard to see where that basement membrane is in that picture. They don't always stain for the basement membrane. So you have to identify where the basement membrane is, differentiate it from other tissues, tell yourself how many layers are there, even if the nuclei are a little bit different than when you typically see them. And you have to see the apical versus the basal shapes and judge from there for just epithelia alone. All right, that's a really good stopping point.